My name is Renee Cottle, and I am an assistant professor in the Department of Bioengineering at Clemson University. Inherited diseases of the liver represents a vast and diverse group of genetic disorders occurring in roughly 1 in 800 live births. In many of these diseases, the underlying cause is a single gene mutation leading to the accumulation of toxic metabolites in the liver and organ failure. One type of inherited liver disease that has received little attention is familial hypercholesterolemia that is characterized by greatly increased LDL levels and a family history of premature severe cardiovascular disease. In 80% of patients, the underlying cause of this disease are mutations that cause the LDL receptors in the liver to be underexpressed or non-functional, hampering the body's ability to clear LDL cholesterol from the plasma. Despite efforts to characterize and treat familial hypercholesterolemia, it remains difficult to treat as many patients do not respond to drug therapies. The only curative therapy available is liver transplantation. However, this procedure carries high risks for mortality and post-transplant complications. The lack of curative therapies has motivated my research on the development of a novel gene therapy for familial hypercholesterolemia. We are proposing to use CRISPR-Cas9 nucleases, a robust gene editing tool, and donor template DNA to precisely insert the correct copy of the LDL receptor gene into the patient's hepatocytes, isolated from resected liver. The genetically modified hepatocytes would then be transplanted back into the patient for the replacement of the diseased hepatocytes with healthy hepatocytes. This approach is powerful because it involves treating the patient using their own cells. In order to replace the native hepatocytes in the tissue, we propose to simultaneously engineer the hepatocytes by silencing the HBD gene to provide resistance to a drug called Sofoba that induces toxicity in the liver, which will provide a selective pressure for the gene-modified hepatocytes. Specifically, our timely investigation will develop new and highly innovative methods to culture primary hepatocytes and genetically edit them ex vivo using non-viral approaches and quantify the extent that they engraft as well as replace native hepatocytes in the liver in a mouse model. Our research represents a high priority and relevance for the advancement of an ex vivo gene therapy for inherited diseases of the liver. With the support of Johnson & Johnson's WiSTEM2D award, I will be positioned to advance in my career as an innovator and leader in the genome editing field while accelerating the development of novel curative cell-based gene therapies for inherited liver diseases.